Welcome to group 3. We are going through lesson 10 group problems here real quick. We're going to try and give our best explanation of these problems, even though a lot of it is vocabulary. But we're going to start with number 1 here.
Okay. Sorry. So I have number 1, and it's saying, suppose a eukaryotic messenger RNA has the following nucleotide sequence. Assume that RNA polymerase II produced this molecule from right to left. And then A asks, which end of the mRNA molecule is the 5' end and which is the 3' end? So it moves from the 5' end to the 3' end. So that means that the left would be the 5' and the right would, the right would be the 5' end and the left would be the 3' end. And then it asks, what is the sequence of the template DNA strand? Include polarity. And so since it's a DNA strand, you're not going to use uracil, you're going to use adenine instead. And so it's gonna, the 5' end is going to be on the left and the 3' end is going to be on the right. And then, should I just go through and say it? It's a 5' A, C, G, A, A, C, G, G, T, 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 A, G, T, A, G, 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 C, T, A, T, C, T, and then 3'. And then, what is the sequence of the coding DNA strand? And so you're just going to go through with the DNA nucleotides, and it's going to be 3', T, G, C, T, T, G, C, C, A, 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 T, G, C, A, T, C, 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 G, A, T, A, G, A, 5'. And then it asks you to draw basically like everything that's happening in transcription. And we have an example right there. And I also drew this one right here. And then the ta-ta box is on the 5' end of the template strand, right? Yeah. And so it's going to be on the left. And so is the transcription start site. It's going to be on the left as well. And that's it.
Okay, my turn. Sorry, it's kind of crazy over here. Hopefully the background noise isn't too loud. So for A, I have a little bit extra information on here. The TATA binding protein, TBP in TFIID binds directly to the TATA box, while sigma factors helix turn helix motif binds directly to the -10 and -35 sequences. And B, guanylyl transferase is a eukaryotic enzyme that links the 5' carbon of one nucleotide to the 5' carbon of another nucleotide. C, group 1 introns use the nucleoside guanosine as a cofactor during catalysis. Am I saying that right?
Catalysis, yeah.
Yeah, okay. While group 2 introns use adenine as a cofactor during catalysis. Cleavage and polyadenylation specificity factor, CPSF, is an endonuclease that assists in terminating transcription in eukaryotes. And for this one, for E, it's TFIIB and TFIIH. I don't want to read that. For F, the spliceosome is composed of subunits called small nuclear ribonucleoproteins, or SNRPs, SNRPs. These spliceosome subunits are composed of 2 components, proteins and uracil-rich small nuclear RNAs. Is that all? And that's all right?
Yeah. All right. So I have number 3. For the first one, it asks about 2 proteins that act as helicases in the prokaryotes or eukaryotes. In eukaryotes, TFIIH acts as a helicase, and RNA polymerase acts as a helicase in prokaryotes. And then asks about one protein prokaryote acts as a helicase to separate RNA from DNA. The real protein does that in the termination sequence, or during termination of RNA or of transcription. And then C asks for 3 molecules in eukaryotes that function as during RNA modification as ribozymes. And it's group 1 introns, group 2 introns, and SNRNPs act as ribozymes. Then indicate one protein prokaryotes that acts as a transcription factor. Prokaryotes called the sigma factor. And then indicate one enzyme in eukaryotes that synthesizes a long strand of identical RNA nucleotides. It doesn't use a template DNA strand for RNA synthesis. Is poly-A polymerase, which is used during polyadenylation, which adds the long strand of adenines at the end of RNA molecules.
So I've got number 4 this time. This one's kind of a fun one. The question's asking you that you hypothesize your karyotype gene called your favorite gene, YFG, contains 4 introns, and you would like to verify the presence of these introns using electron microscopy method, drawing images of the resulting nucleic acid structures following the hybridization experiments describing parts A through D alone. So I'm going to try and go through this. I've got 2 pictures for you, one from the slides as well as one of my own personal pictures, so the slide one's probably better. Let's get this going here. So here are the questions. I'm just going to go through each one. The pre-mRNA for your favorite gene is hybridized to the genomic DNA copy of your favorite gene. So this is kind of my DNA template strand. This is my pre-mRNA right here. And so my DNA template strand. You can also see it here. So this would be your template strand, your coding strand, your pre-mRNA comes from that, then plus your mRNA, and so your mature mRNA binds to that and creates your intron. Back to the question. So that's that one, that first one, which is your 2. Then the mature mRNA for your favorite gene is hypothesized to the genomic DNA copy of your hybridized to the genomic DNA of your favorite gene. So that's where this one would be in. So this is where the hybridization comes from. The introns get popped up into these intron loops, and your mRNA is sitting right there on top of your template strand. This is what it looks like right here. And then we're going on to number C. The mature mRNA for your favorite gene is hybridized to the genomic DNA copy of your favorite gene. This is, in this case, well, pretty much this is what's going on right here from what I understand. And so your coding strand is hybridized to your template strand right there. So that's what's asking, right? Hybridized genomic DNA. Your mRNA is hybridized genomic DNA. This is in terms of removed using the splice site method. And then the last one, the mature mRNA for your favorite gene is hybridized to the genomic DNA copy of your favorite gene. In this case, 2nd and 4th introns in the pre-mRNA both contain defective donor site, and the branch site is and the branch site editing assume that introns are removed using the splice site method. And so this is what I did. Kind of had my genes here, had my lariat come through, removing of the splice design. So I had 4, removed 1, and now I have 2 or 3, and the 2 got removed. And it kind of works like this. And then what happens is some SNRPs come in, U1 through U6, and they bind together and splice these suckers off, and that's how you get those. I hope I explained that well enough. It gets kind of confusing there at the end. Definitely look at the slides, definitely look at the video if you want more help on that one. So.
All right. Then I have to provide a list of proteins, nucleic acids involved in transcription RNA modification. And letter A asks which of the above is found in the bacterium Escherichia coli, so E. coli. And it's NUSA, RUT, and -35. B asks which of the above is found in the yeast, however you pronounce that.
Saccharomyces cerevisiae.
There you go. Enhancer, -25, group 1 intron, TFIID, SNRNP, silencer, and GC box. C, which of the above is involved in transcription initiation, is -25 enhancer, -35 GC box, TFIID, and silencer. D, which of the above is involved in the transcription termination, is RUT and NUSA. And which of the above is a DNA sequence, is there's -25 enhancer, -35 GC box, and silencer.
Great. So that's lesson 10, group problems. Hope we had enough explanation there, and have a good night.